Okay, it's a great pleasure that I welcome my dear friend and colleague, Dr. Rajendra Apte, as this year's Snell Memorial Lecturer. Dr. Apte is the Paul A. Sibis Distinguished Professor of Ophthalmology and Visual Sciences at Washington University School of Medicine in St. Louis. He also serves as Vice Chair for Innovation and Translation and holds professional roles in the departments of Developmental Biology and Medicine. He's a renowned vitreoretinal surgeon and clinician scientist whose groundbreaking research has significantly advanced our understanding of retinal diseases. His work focuses on the molecular mechanisms underlying conditions such as age-related macular degeneration, diabetic retinopathy, and retinal vascular diseases. This includes how immune dysfunction, aging, and lipid metabolism contribute to retinal degeneration, leading to novel therapeutic strategies. He has published more than 200 peer-reviewed journals, articles, book chapters, and abstracts. His contributions have been recognized by the American Society of Retina Specialists and the President's Award. Most recently, he's been recognized with the Prion Award, which he'll be receiving this June from the American Society of Retinal Specialists. Dr. Apte attained his medical degree from the University of Bombay, followed by a PhD in immunology at the University of Texas Southwestern Center, and he completed his ophthalmology residency at UT Southwestern, pursued a vitreoretinal fellowship at the Wilbur Eye Institute. And here's a picture of us. He was a co-resident of mine. This is the Retinal Vascular Center in 2002. And you can see me on the far side there. And then Dr. Ghazi's between us, but there's Raj, looking very young. He always looked very sharp. And you can see our co-resident Yellow Marguerite. And in the background, there's Andy Shackett, Sharon Solomon, Neil and Susan Bressler, and Dan Finkelstein, who were our mentors there. Not pictured there as Boss Sada, who was also there as a new faculty member that year. That was a great time. And he was a phenomenal colleague. Since joining Washington University in 2003, he's become a tenured professor and has mentored numerous emerging leaders in ophthalmology. Please join me in extending a warm welcome to Dr. Rajendra S. Apte as he presents Aging, Inflammation, and Metabolic Processes Involved in AMD Pathogenesis and Developmental of Novel Therapeutics.
Thank you, David, for that generous introduction. And I was looking at that list of people, and I'm still wondering if that was the wrong person that got the email. So I'll try to do justice to the Snell Memorial Lecture. So I was at the FLOM in 2005, so it's been 20 years since I last presented here, and so I'm going to try to present what we've been doing in the last 20 years. I have a number of disclosures related to just the science and the intellectual properties. And I'll focus, just for the sake of a cohesive story, most of the work on understanding processes and how I can relate them to macular degeneration, but they relate to a number of retinal diseases. And why is science important? Since, as we know, there's a lot going on with science and science funding. To give you an idea, when David and I were fellows, there was no anti-VEGF. So, and that was 2001 to 2003. So things changed fast, and they only changed because someone cloned VEGF in 1989. And so, it takes time for discovery to translate. So this is the life cycle of macular degeneration, which I don't need to tell you about, but just remember, we have focused a lot of attention here, rightly so, on atrophic disease and neovascularization. But disease starts before we can see it because there's a lot of genetics and other factors that go into it. And then we identify it when we have these lipid-rich deposits. So what I'll try to do is do it sort of the Benjamin Button way, going backwards, how we focused on molecular processes here and how we know a lot now to where we can translate this into future therapies. So when I started doing this, at least at WashU in 2003, there was a lot of attention focused on immune cells and how they regulated pathogenic neovascularization, and a lot of attention on macrophages, both resident macrophages, which we call microglia, and then monocyte-derived macrophages. And what we found, and all of these CNV models are in mice, using the injury-induced neovascularization, and looking at kind of the volume of pathogenic vessels we get. And we found that macrophages were actually quite good at inhibiting neovascularization. When you adoptively immune transferred them intravitreally into the mouse eye, but T cells that are CD3 positive or dendritic cells that are CD11C positive were not anti-angiogenic. But this was not necessarily the findings that everyone was getting, that macrophages we knew could also be pro-angiogenic. And then a lot of work from a number of groups, including ours, showed that the flavor of the macrophages was important, whether they were activated in a certain way, polarized in a alternatively activated manner or a classically activated manner. The age of the host was important. So if you derive macrophages from old animals, they lost these anti-angiogenic properties, and they could also become more pro-angiogenic. So there were a number of molecular pathways that were important. I'm showing you one here. If you had hyperactivation of the JAK-STAT pathway within these immune cells, then it made these cells a lot more pro-angiogenic. And so that has been shown to be important now in macular degeneration. There were some initial efforts at targeting these pathways to influence neovascularization in a VEGF-independent manner. And over the last 20 or 25 years, this is no longer new news, because macrophages in different tissues, they even have different names, right? And they do different functions depending on how they're activated, what the microenvironment is, and kind of what niche environments they last in. They can be inflammatory, they can be pro-angiogenic, or they can be regulatory. I tried to coin a new phrase for macrophages in the choroid and the retina called IMAX, but I think the name was taken, so I didn't have much success. So what I'll try to do in the next 30, 40 minutes or so is tie in how immune cells that are present in homeostatic conditions and in disease interact with what happens with dysregulated metabolic processes. And in AMD, an important dysregulation is in lipid metabolism, which is why you get these, pseudoreticular drusen or subretinal deposits, sub-RP drusen, and other forms of lipid deposits. And then sort of tie that in again into things that are going on with aging. We know that genetics is very important in AMD, and at least 4 genes in the lipid pathways, the pathways that regulate lipid metabolism, are important. I'll talk a lot about ABCA1, which is on here, but then the other genes such as CTP, lipase C, et cetera, are involved in processing of cholesterol and lipids from tissues back to the liver. Because normally extracellular lipid is a danger signal, whether it's in atherosclerosis or in the eye, and you don't want extracellular lipids sitting in tissue, so it has to be transported back to the liver for excretion in bilirubin, in bile and in the stools. So we know that there is an increased risk when you have polymorphisms in lipid pathway genes, and we wanted to explore this interaction between lipid-related genes and neovascularization. So that's where we started the work with a couple of postdocs in the lab. What we found initially was that just like humans and human patients with AMD, age is important. So if you had old mice, and for definition of old in mice, it's older than 18 months, they were prone to get larger neovascular complexes when you compare them to young mice. So there's something about the microenvironment in old mice that made them more prone to larger CNV lesions. Similarly, if you took young mice and you put them on a high-fat, high-cholesterol diet, they started behaving like old mice where they got much larger CNV complexes. So having a higher lipid load made the phenotype of CNV similar to what we saw in older animals. Again, telling us something about this relationship between lipid metabolism and the propensity for developing neovascularization. So what we did next was took a couple of these genes, these ABC transporter genes, and I'll talk a little bit more about them in a minute. These are transporters on the surface of a lot of cells in the body that actually allow cells to kick lipid out. So all of these apolipoproteins like HDL can then transport this cholesterol and lipid back to the liver. So when we knocked out these genes from myeloid cells, so microglia, macrophages, it's been done now, and I'll show you in rod photoreceptors as well as RPE cells. What we found was this really interesting finding that when you just knock these genes out in microglia and macrophages in the retina, you had, again, a very similar phenotype, similar to old mice and similar to mice that who were on the diet-induced obesity model for about 3 to 6 months, telling us that these transporters that normally took extra lipid back to the liver were important in regulating pathogenic angiogenesis in the eye. What was also equally exciting to us is that this was a pathogenic mechanism that was therapeutically modifiable. So if you took boosters of ABC transport expression, so if you took something called a T compound or LXR agonist that bump up expression of these transporters, we could, in a dose-dependent manner, either given systemically, that's shown up here, or locally in the form of eye drops, reduce the CNV burden in these mice, telling us again that we can imagine a day when we can improve the carrier capacities in patients, and maybe, I mean, the big goal is to prevent AMD, but we can prevent conversion from early or intermediate AMD to these more advanced forms of AMD, such as neovascularization or atrophic degeneration. And the reason that's important is because we know that if patients sit at an intermediate AMD phenotype, they don't necessarily have vision loss. Vision loss is typically associated with the more advanced forms, as you know. So I'll leave neovascular AMD here at least for a little bit, because we were very interested in how this dysregulated cholesterol metabolism pathways were involved in the atrophic forms of AMD. Did it actually, if you're dysregulated lipid homeostasis, did it actually allow a cholesterol and lipids to accumulate in the subretinal space? And that's what I'm showing here. This is the same model where we knock out the ABC transporters in myeloid cells. And if you age these animals, so you're not doing laser here to induce neovascularization, you're just aging these animals. And these are the 12-month-old mice. But if you look at 6 months, all you see is sort of mild scotopic A-wave dysfunction and dark adaptation problems. And if you know patients with intermediate AMD, even when they have 20/20 vision, many of them, those are the two things they show functionally, right? They have impaired dark adaptation and impaired early rod responses, because it's the parafoveal rods that are affected very early in macular degeneration. But as these mice age further, you can see that they have scotopic as well as photopic dysfunction. And then you have these subretinal deposits that you see by 6 to 12 months of age that keep getting worse. And by 18 months of age, they actually have pretty severe neurodegeneration. What happens if you knock out these transporters out of the rod photoreceptors? You have a phenotype that is, again, phenocopies some features of AMD, but is slightly different. So you get increased RPE lipid droplets. And in humans, you don't get droplets, you just have increased RPE lipids. And then you also get outer retinal vacuolization in the photoreceptors, and again, a neurodegenerative phenotype. And again, a couple of postdocs in the lab and a graduate student were really involved in showing these phenotypes. And then we, and also Christian Grimm in Germany, knocked out these transporters out of the RPE, and you get a very, again, severe form of lipid accumulation in the subretinal space. So we learned a lot from these initial experiments of altering the ability of the outer retina and the RPE to handle lipid. So when we influence this process in a way that these cells could no longer transport these lipids well, we got these various phenotypes that recapitulated certain features of AMD in humans. Even though we all know that mice really don't have maculas, we can create features of early and intermediate AMD. A number of factors, I have this sort of cartoon slide up here because a number of factors influence this. Of course, aging influences this. I talked to you about the ABC transporters. There are a number of microRNAs that I won't show you the data, but I'll just touch on them. There's microRNA 33 that regulates the expression of these ABC transporters. And with age, the expression of this microRNA goes up pretty dramatically. And so that's what downregulates the expression of these transporters with aging. There's a fatty acid desaturase that's regulated by miR-150 that also influences the ability of myeloid cells and microglia to regulate angiogenesis. And there's another microRNA we discovered, miR-34A, that regulates a transcriptional factor called KLF2. So there's a number, and that regulates choroidal neovascularization in a VEGF-independent manner. The reason to mention this is this opens up sort of possible therapeutic avenues that we can deal with VEGF-independent processes, because even though we have sort of a magic bullet in VEGF antagonists to treat neovascularization, we know that we have a lot of problems with incomplete response, high treatment burden, and persistent or recurrent neovascularization. So we do need additional agents to help us with this treatment load. So in our lab, there were two graduate students that were working on a completely independent project related to metabolism. And this was really involved with how organisms age. And they were working on a molecule that we all study in biochemistry called NAD. I mean, NAD has redox functions, but they were really more focused on the coenzymatic activities of NAD. So NAD works as a coenzyme in a number of enzymatic reactions, including 3 in the Krebs cycle. So it's very important in mitochondrial function and energy generation. And so what they showed was that this process was really important in aging. So as humans get older, the levels of NAD, intracellular pools of NAD go down pretty dramatically. So if you compare a 25-year-old to a 50-year-old, a 50-year-old NAD levels in these pools is about half a 25-year-old. If you take NAD out of a cell or out of an organism, the organism will die in basically 30 seconds. So it's a critical molecule. And we showed several years ago that this process was really critical in rod and cone mitochondria, where the coenzymatic activities of NAD and the biosynthetic pathway regulated by 2 critical enzymes. I'll talk about NAMPT a little bit, but which converts the precursor called NMN into NAD was very important. And you disrupted this either pharmacologically or genetically, photoreceptors died. And I'm mentioning this because it sort of ties into how we identified some of the alterations in myeloid cell function and in lipid dyshomeostasis. So I'll take you back to the model where we disrupted these transporters that I talked to you about, the ABC transporters that regulate the ability of the RPE and other cells to process extra lipid, use apolipoproteins, and take it back to the liver so it can be pooped out, basically. And then when you didn't have this ability, as I showed you, you had lipid deposition and neurodegeneration, similar to what we see in intermediate AMD and in advanced AMD, like geographic atrophy, although mice don't really get actual foci of GA, they get more of a retinal degenerative phenotype. So as we further characterize what's happening in these retinas in mice that are lacking these transporters, and you get these subretinal lipid deposits, we found that these cells were actually senescent because you can see in the green over there with the arrows that they are beta-gal positive, and they also have lipofuscin, which again can see in AMD and also in other inherited retinal diseases, as you know, like Stargardt's. We know these cells are myeloid cells because they have the markers for F480 and EBA1, and they are clearly senescent because they have, in addition to beta-gal, they're spider-gal positive. So a couple of the talented postdocs in the lab came up with this experiment. They said, can we recreate this disease by actually injecting myeloid cells that are either normal or that are missing these transporters? And so they just cultured them in a dish. And when they injected these cells intravitreally, nothing happened. So you could inject, wild type cells or the disease-producing cells, let's call them, intravitreally, and we didn't get a phenotype. But when we injected them subretinally, only when you injected cells that were deficient in the transport capacity, you recreated, again, both fundoscopically as well as with OCT, of a phenotype that looks reminiscent of pseudoreticular drusen in AMD. It's not identical, of course, because the lipid composition is different, and it's a very cellular phenotype. And these cells are lipofuscin-rich also. Interestingly, these cells are clearly senescent because both by Sen-Mayo markers as well as expression of P16, P21, and some of the secreted SASP factors, you can show that these cells are not just like old cells, but they're like senescent cells. And we know that senescent cells accumulate with aging, and they're associated, even though they're a protective mechanism against development of cancers, accumulation of senescent cells with aging that are not associated with sort of this cancer protective phenotype can be bad for many reasons because they create a pro-inflammatory disease-producing environment. And so when we tried to characterize these cells by bulk sequencing, we were really excited because we started seeing some of the genetic signals that I tried to show you earlier. So when we compared normal to the disease-producing myeloid cells, one of the top candidates I already told you we knew about is a fatty acid desaturase. But then the second highest hit was CD38. And if he hadn't been working independently on NAD metabolism, we would have just totally missed it. Because CD38, which was overexpressed in these disease-producing cells, is a consumer of NAD. And so it's an NAD degrader. And so we were, of course, very, very interested in finding out that although we had already shown that the biosynthetic capacity of NAD is reduced with aging, now we were finding out that one of the things that degrades NAD was also upregulated, both in the genetically deficient mice, and then also we've shown it before in old mice. So tying in sort of this well-known pathway that's associated with aging and disease to this dysregulation of lipid metabolism. So I'll just show you a few lines of evidence. I think I can skip this slide, but just showing you that before what I showed you is if we inject these disease-producing cells in the retina, we can produce an AMD phenotype. Here we did something different. We knew that these cells were overexpressing CD38. So here we just overexpressed CD38 in otherwise normal myeloid cells and then injected them in the subretinal space. And here, by just this one difference, expressing CD38 in a myeloid cell, again, reproduced or recapitulated the phenotype that we see in the ABC transporter deficient animals. So showing you that you could either have impairment in lipid metabolism or the effect of that, which is CD38 overexpression, that would give you an AMD-like phenotype. So the working model here is that cholesterol overload is driving up the expression of the LXR receptors transcriptionally, and that's good because that usually drives up the ABC transporter expression, which allows you to kick out this excess cholesterol load from the outer retina as well as the RPE. Because remember, every day with the diurnal rhythm, we're having like about a tablespoon or two of lipids, right, that the body has to handle to clear these outer segments. But inadvertently, and we don't understand fully the mechanism for this, the upregulation of LXR is also associated with driving up of this NAD degrader called CD38 that then reduces the pools of NAD, which is critically important in the health of these cells and is driving these myeloid cells or RPE cells into senescence. And that's an unhealthy phenotype that's creating sort of the microenvironment, both for atrophic disease and for neovascularization. The last therapeutic avenue is, you can either downregulate the expression of CD38, you can remove lipids, or how about augmenting these metabolic precursors that we need to maintain a healthy outer retina and RPE. So what we did here was we showed that if you take the precursors of NAD called NMN, and if you take the knockout mice, the ABC knockout mice that I showed you before, and you just give them NMN systemically, so you just administer it to them either intraperitoneally or in the water that the animals drink, then you can reverse the disease phenotype. You can reverse the expression of these senescence markers, and then you can improve the mitochondrial function, as we tested here by seahorse, just by giving addition of nicotinamide mononucleotide. We can improve the respiration of the mitochondria in the microglia and the RPE. And then the last thing you could do is you could just clear these senescent cells by using either genetic or pharmacologic approaches called senolysis. Unfortunately, we had a recent senolytic drug, as you know, a BCL-XL that they studied in a phase 2 clinical trial that didn't give positive results. But I think this is still an approach that's rational if you find the right disease stage and the disease target. So here's just showing you that, again, in a genetic model of senescence, if we if we cross our transporter mice to a construct where we could pharmacologically give a dimerizer, we can basically reverse the disease. We can show that we can clear subretinal deposits. We can reduce the features seen both fundoscopically as well as by OCT, and then we can improve the, not completely, but at least partially improve the visual function in these mice. So this tells us that, we don't have to think of just preventive approaches in AMD. We actually have a possibility of intervening early in disease and sort of reversing the cascade of impairment. So I'll skip the pharmacologic approach to senescence for the sake of time, but this is really showing you that with the combination of D and Q, you can pharmacologically remove senescent cells and also have improvement in visual function in these mice. So again, here's a model of how we can now think of AMD as a disease process. So what's shown here is the left is basically normal, right? And on the right, what you're seeing is that there's a lot of cholesterol in the lipid that the eye sees, and especially the macula sees all the time. And what that does in normal physiology is triggers this LXR response so that you can have ABC transporter upregulation so the retina can handle this lipid so it can be removed with apolipoproteins through the choroid. But with aging and a number of other things like genetics, risk factors like smoking, you have impairment of multiple of these processes, decrease ABC transporter expression, increase CD38 expression that leads to reduced NAD, mitochondrial dysfunction, and senescence. So even though all of these processes are impaired, you can imagine that each of these is therapeutically modifiable. We can have apolipoprotein targets, we can have NAD boosters, and then we can have senolytic clearance mechanisms by which we could intervene very early in AMD to prevent sort of this progression, because you've sort of got this slow coal fire, right, that sits there for years and years and years before you develop geographic atrophy or choroidal neovascularization. And right now we do have, fortunately, some agents to treat these. But, just as I said, we didn't have VEGF antagonists in 2003. I'm hoping that we won't need complement inhibitors or VEGF antagonists in 2042, because we might have things that we can intervene much earlier in the disease cycle. And I'll tell you one approach that I'll show you next that we are doing that will really pharmacologically allow us to remove excess cholesterol from the eye. So again, just remember the disease cycle of AMD. At least we can be ambitious enough to say we can intervene here in the disease. There's a number of clinical stage programs right now that are doing that. One of the ones is those cyalomimetics that are trying to improve the macrophage activation to, again, allow reduce inflammation and improve lipid clearance so that you don't progress to at least the atrophic or neovascular stages of disease. But one of the things that I've become a firm believer in is that this lipid accumulation or cholesterol accumulation you see in these various forms of laminar or linear deposits in AMD, it is upstream of everything else. It's upstream of complement activation, mitochondrial damage, inflammation. And so I consider that the root cause of AMD before you get photoreceptor RPE damage or before you get neovascularization. And I'll spend a few minutes just talking about something we've discovered recently that I'm very excited about, that is a way of designing custom apolipoproteins to target these intermediate stages of AMD. So apolipoprotein M it's a new apolipoprotein. It's not as well understood as some of the other ones, but it sits on HDL. And it's a chaperone for a sphingosphene 1-phosphate that has 5 different receptors, 2 of which, receptor 1 and 3, are expressed in the RPE. And it has got some very interesting epidemiologic associations. So ApoM is inversely associated, levels are inversely associated with heart failure mortality. And it strongly appears again, inversely correlated as a link with diabetes and heart failure. And then low levels of ApoM are associated again with high mortality in right heart failure. So there's clearly a protective role that ApoM plays in cardiovascular disease. And so when, for one reason or the other, we got involved in ApoM research, and in collaboration with Ali Javaheri at WashU and then Christina Christofferson in Copenhagen, what we found in a large cohort of AMD patients was that their serum levels of ApoM were very low. And we know that AMD patients, and there are multiple studies that have shown that they have serum dyslipidemias. They're broad serum dyslipidemias, but the relationship is very complex because, as you know, the genetic variants that are risk alleles are HDL elevating. So it's like the opposite of cardiovascular disease, right? So we know that the genetic associations are complex systemically. And so I think what's really important is what's going on in the outer retina and the choroid in terms of how we modify cholesterol handling and cholesterol carrier capacities. But we were interested when we saw this result that ApoM levels were significantly lower systemically in AMD patients. So what we did was we again took our ABC knockout mice that on a high-fat diet get these apolipoprotein, these AMD-like phenotypes, and we just treated them. We either treated them with transgenic plasma, ApoM transgenic plasma, from mice that basically produce 5 to 15-fold higher ApoM, or plasma from knockout mice that didn't have any ApoM. And we saw a significant improvement here in the turquoise RPE function. So this is the light bleach recovery of the RPE. And so you can see that just giving them ApoM-enriched plasma improved RPE function. And if you look at it by EM, you can see that the lipid droplets that accumulate in the RPE in these mice improved fairly dramatically and were associated with as I showed you, an improvement in the visual function of these mice. As I mentioned to you, ApoM binds with, is a chaperone for sphingosine 1-phosphate, and we wanted to see if this binding reaction was important. So what we did here was, again, a simple experiment where we created two types of mice. One is just a regular mouse that this binding capacity was normal, and then a mutant mice where there was a point mutation where the ApoM just couldn't bind the S1P. And as you can see here, we did, again, the similar therapeutic experiment and showed that if you use the point mutant mice, basically compared to the mice that had plasma that had the point mutation, you had a therapeutic effect if you gave them the control. You lost the therapeutic effect compared to the control mice where you had the ability of the ApoM to bind S1P. So this S1P ApoM binding interaction was important in this improvement in visual function. And again, if you lost that binding capacity, as shown on the right, you didn't have an improvement in the lipid droplets. So again, you had a lot of lipid droplets in the RPE because the ApoM now couldn't function to remove the lipid as it was doing there. So again, we didn't quite know exactly how it was doing it. The ApoM S1P complex binds the S1P receptor. So we did single cell sequencing to identify that there were 2 receptors on the RPE that were expressed, 1 and 3. And to make a long story short, if you were missing, this is a whole body knockout that we made. If you were missing the receptor 3, even though if you gave the mice, even if you didn't therapeutically do anything, if you just compared the wild type mice to the S1P receptor knockout mice, they had a defect. So these receptor 3 knockout mice already had high levels of lipid droplets and RPE dysfunction. And so again, it tells you that this ApoM S1P and S1P receptor binding pathway is really critical in the ability of the RPE to prevent lipid accumulation. The last thing I'll show you here is that how is the RPE doing it intracellularly? What's the molecular machinery that's involved? And what's showing you here is that when we knocked out this gene called LIPA-A from the RPE, it made a profound difference, because when we knocked this out, even though the rest of the pathway, the ApoM, the S1P receptor were normal, the RPE was not able to use the lysosomal clearance mechanisms to remove the lipid. And so again, the lipid accumulated in the RPE, despite all of the other processes being normal. So we feel that we completely understand this process very well now. And we think what's happening in the outer retina is that these apolipoproteins like HDL use ApoM on there to help the RPE clear the normal lipid loads. And in AMD, what's happening is you have inherently it's not the only thing that's happening, but you have inherently very low levels of ApoM and probably some other apolipoproteins that are not allowing the RPE and the outer retina to function well. And that you can imagine over 7 decades of life in people with genetic risk and other factors, allow you to accumulate lipid in the subretinal space. So this has actually led to a preclinical effort that we have started to design custom apolipoproteins that we have now tested in mice, rabbits, and non-human primates. And we're really excited about this possibility of taking apolipoproteins that we can control the peptides and we can control the lipid cargo and basically give fat to remove fat, because that's how lipid transport works. And by doing this, we're hoping in the next 5 to 10 years, we can design hoping in the next 2 to 3 years clinical trials, but maybe in the next 5 to 10 years, have therapeutics that are apolipoprotein based that will allow us to treat AMD much earlier than waiting for neovascularization or waiting for geographic atrophy to then treat it. It doesn't have to be an isolated approach, right? We know we have AREDS2. We're going to have other things in our armamentarium. So it's very feasible that treating AMD earlier may involve some type of synergistic or combination approaches. But I'm just knowing the molecular pathogenesis of this now, I'm not going to one in favor of sort of systemic approaches because I think the off-target effects there are too large. And so if we can design locally delivered therapy, the cargo requirements are very small, and we can, in a safe and a very effective manner, develop agents that can help us clear this phenotype, which is drusenoid deposits, which is how we diagnose patients with AMD, and prevent vision loss from complications that we see further downstream. And so I'll sort of end here given the time. But again, just wanted to highlight, some of the work we've done, tying in immune mechanisms in aging, dysregulated lipid metabolism, and the energetic requirements, and how they all interact with each other to create a pathogenic environment that causes biomicroscopic manifestations of AMD. But by understanding that, I think we have opened up multiple avenues of therapeutic interventions that we don't have right now, but that are at clinical and preclinical stages of development. So the NAD boosting possibilities that I discussed with you, there are actually 5 clinical trials we're doing right now in various neurodegenerative and mitochondrial diseases to see if we can actually improve phenotypes there. And the hope is that we can eventually design a trial for retinal degenerative conditions like RP or AMD to where we can combine these approaches to treat things much earlier than what we've been able to do so far. So again, thank you for the invitation, and happy to answer any questions.
Questions? I'll start. I'm just curious on a quick, simple question. Do we know if there's difference in macrophage type between retinal neovascularization versus choroidal neovascularization?
Yeah, so now, the field is becoming very sophisticated. And so there's a number of people working on sort of, using spatial transcriptomics to kind of genetically characterize this. So Jeremy Levine from Northwestern just recently published a paper on a spatial map of macrophages in choroidal neovascularization. I don't think he really has all of the different subtypes, but he can find that in various parts of the CNV lesion, you have a different transcriptome associated with the macrophage. So clearly, there are definitely different subtypes depending on kind of where you look.
Yes, you mentioned 5 clinical trials.
Yeah.
Sir, are those animal studies or human, and are they directed particularly at STD or other conventional forms of drusen, and what sort of delivery?
Oh, yeah. So the clinical trials on the NAD precursors, that's a great question. Sorry, maybe I misspoke. Those are all clinical trials. They're registered, so you can look them up. But all of those phase 1 and 2 clinical trials right now are in non-eye diseases. So it's Alzheimer's, Friedreich's ataxia, acute kidney injury, AKI from cardiovascular disease, because the applications of the effects of NAD metabolism and mitochondrial function are pretty broad. And so the hope is we can design an AMD trial that targets geographic atrophy, basically the prevention or growth rate or reduction in progression of growth rates. The delivery of that is oral. So that's an oral agent because the bioavailability and the safety record for that is actually quite good. And so that's the one that's not an intravitreal approach. But there's not an active trial right now for that. That's sort of in preparation because of design and cost concerns. So.
Oh yeah, Raj. So great talk, Raj. That's a lifetime of work that you have presented. Congratulations on what you have done. I have a question based on a lot of clinical trials looking at removal of drusen by laser, or micropulse laser. There have been multiple systematic reviews showing that disappearance of drusen, while they were effective, the laser, micropulse laser, but they did not stop progression of either atrophy or the occurrence of choroidal neovascularization. So how do you look at this dichotomy of drusen deposit leading to this versus when you remove those drusen, the AMD still progresses?
Yeah, it's a really good point. So with the laser, of course, there's a lot of caveats, right? I mean, you're sort of doing my feeling is what you're doing there is with the subthreshold or whatever thermal injury, you're activating the glial cells. So there's probably glial cell migration, and the idea was maybe they're phagocytosing debris and material. But that may not be the best way because the polarization that you're achieving of these cells is probably actually activating their undesirable functions, so to speak. But if you think of removing excess cholesterol, right? I mean, if you do it chemically, I mean, it's not the perfect example, but the best example would be athro, right? With athro, you're not trying to sort of hit it with a laser to clear the atherosclerotic plaque. I mean, the whole purpose is to prevent plaque burden and then the inflammation that's associated with it. So my feeling with any kind of lipid-based approaches is that if you use as close to the natural mechanisms, which would be, can you take what the body normally uses, which is apolipoproteins, right, to pick up excess cholesterol and then take it out of the eye? That's a much more viable approach. And if you do it early enough before, you get all of the drusen collapse or hollowed out drusen, because I think at that stage, you've already started the damage pathways. So if you intervene early enough and your surrounding tissues are healthy, then you have a much higher chance of rejuvenation because you've not, induced sort of these death pathways already, whether it's GA or whether it's RP damage and neovascularization. Of course, I mean, we would have to do the clinical trials to actually know if that works, but at least based on the cellular cell culture models and animal models, that's what it suggests, at least that's the way to go.
Fantastic. Thank you so much.